So thank you very much, Thomas and Alex, for the kind invitation, and I'm excited to present the talk. So the topic, my topic for the next generation cytogenetics session is the optical genome mapping. So here are my disclosures. None of these are relevant for this particular talk. So keeping in mind the educational format of this session, I've kind of broken down my talk into 6 separate topics. So I will start with the basic principles of the technology and the simple workflow and the sample requirements, how to analyze the findings and interpret for clinical reporting, as well as discuss the challenges and limitations. And followed by that, I'm going to step up the game to kind of highlight the potential clinical and research applications of this technology, and finally summarize it and give perspectives on future directions based on the potential. So first, let's begin with the definition and principles. So I guess to this audience, this slide is not anything new, so I'm not going to dwell on this. But it is fascinating to kind of live in this era where there is all the advances in the techniques to identify the structure variants. Now, these have been largely overlooked for clinical profiling. We've focused on single base pair level next generation sequencing in the past decade that has thrown insights into several of the cancers and constitutional disorders. And of course, we've been doing conventional karyotyping and FISH, as well as microarray. But now we have the optical genome mapping, whole genome sequencing, long-read sequencing that are going to open up a lot of potential for patients. So what exactly is optical genome mapping? So this is a technology primarily that can be used to assess genome-wide structure variants at a very high resolution, often at a gene or an exon level, and we can go down to 500 base pair level. So it does that basically by evaluating the changes in the fluorescent labeling pattern, which is specific to a 6 base pair sequence in each of the DNA molecule by comparing it to the reference. So essentially, this would be a high-resolution karyotyping. Right? So the labels, the fluorophore labels, are introduced enzymatically without really damage to the DNA backbone using the DLE1 enzyme. And as you can see here, it is specific to the CTTAAG sequence. Now, this sequence is present ubiquitously in the entire genome. And everywhere there is this sequence, the fluorophore goes and inserts itself. Now, this is great because in about 100, on average, about 100 kilobase pair sequence of DNA contains about 14 or 17 of these markers. So followed by labeling, the DNA are linearized through nanochannel arrays. And then once they are linearized, the fluorophores, the labeling pattern is almost like a barcode. And we can match up this barcode to the reference, using the reference genome to find out where it came from. So as you can think, the higher the number of labels, higher the uniqueness of that barcode, and higher the accuracy. The real key for the optical genome mapping technology, the highlight of this technology, is the use of this ultra-long high molecular weight DNA. So on average, it's about 300 kilobase pair. So that allows the preservation of these labels relative to each other that gives the technology the higher confidence levels to interpret structure variants. It is also important to remember there is no amplification. It is in the OGM. And like I mentioned before, the labels are present, are distributed throughout the genome. It could be variable, but overall ubiquitously throughout the genome, and so there is no enrichment of the targets. So again, I want to emphasize some points over and over. So this is the high molecular weight DNA that allows the generation of the contiguous maps without interruptions, large maps. So these are especially good for detection of structure variants. Now, we all know the structure variants are often flanked by numerous repetitive regions. And because of the use of this high molecular weight DNA, we don't lose that information in this technique. So that allows it to generate the data with a very high accuracy, specifically for SVs. Just to give you an idea, the length of the molecule is pictorially represented here, what is used in the OGM. So this is this is the DNA extracted from FFPE specimens that are completely fragmented. And here is the column-based DNA extraction. And we can see how long the molecules, the DNA molecules are for OGM technology. We are aware that there are other technologies, right, that can be used for detection of structure variants. Primarily the sequencing-based ones. So OGM is the non-sequencing-based ones. There are some inherent limitations to using sequencing-based techniques, as I mentioned before, primarily because of the repetitive sequences. So in addition, these sequencing-based techniques are also able to identify the mutations, but because OGM uses ultra-long molecules, it cannot go down to the single base pair level. So we're not able to extract the mutation information. But considering that, for example, in hem-onc specimen, targeted NGS is almost a routine practice. So combining the OGM, high accuracy structure variant data from OGM with the regular short-read NGS, targeted NGS at a very deep coverage level can provide a great insight into what's happening into the genome. See, so this slide kind of highlights the amount of the data generated by a typical OGM run. So the technology of OGM allows routine processing of these large DNA molecules. So as I already mentioned, about 300 kilobase pairs, ranging between 200 to 800 kilobase pairs. In comparison to long-read sequencing, this is about 10 to 30 kilobase pairs. Now, these are assembled to almost 70 megabase pair maps, overall generating about 5,000 gigabase pair data per flow cell. And that allows the mapping of the entire chromosomes at a very high resolution. So again, as I mentioned, the preservation of the structure and orientation of these labels in the genome is what gives it the high confidence level for SV detection. So with that, let's move on to the workflow and sample requirements for optical genome mapping. So we can kind of divide that into 6 steps: DNA isolation, labeling, linearization, followed by imaging, conversion to digital data, and analysis. So I'm just going to highlight the key points that are relevant or unique to this technology for each of these steps. So for the DNA isolation, so as I mentioned before, the DNAs required for OGM are extremely long. So there is a specific technique that is required. We cannot use the regular archived DNA that we routinely extract for NGS. So it uses a paramagnetic disk that traps the DNA so that during the wash cycle, the shearing forces that kind of break the DNA apart is minimized. So it is important to note that there is a special skill required to extract the DNA. It can be done. It's just a matter of a learning curve if you're used to extracting the DNA for other technologies routinely. So it is especially important when pipetting. It has to be slow and carefully done, so there is no introduction of bubbles that might break the DNA apart. So overall, like on average, about 300 kilobase pair DNA is obtained. At this point, let's also think about what kind of samples are pertinent for OGM. So the acceptable samples are bone marrows and peripheral blood, especially relevant for hem-onc, right? So as the mononuclear cells within these sample types, the primary cell cultures, immortalized cell lines, organoid bodies, for prenatal testing, amniotic fluid, chorionic fluid. So essentially, any cell, if the cells are intact, to obtain the intact DNA, it's usable. And for OGM technology, about 1 to 1.5 million cells would be an ideal number. We can use either fresh sample or even frozen sample if the cells are cryopreserved with viable cells at -80. I personally have used samples as old as 5 to 6 years, and they worked fine. The unacceptable samples, of course, FFPE samples, because of the excessive DNA fragmentation, cannot be used for OGM. DNA that has been extracted for routine sequencing technologies cannot be used. These are too short. And sometimes, if the cells are preserved for over a couple of weeks at -20, this is unacceptable as well. So let's move on to DNA labeling. This I already mentioned before. I'm just going to reiterate just because it's important. So the labeling is done using enzymatic technique and not based on nicking. So it doesn't damage the DNA much. So it is done, so there is covalent modification of the CTTAAG sequence. And as I mentioned before, about 14 to 17 labels. So here is, so now you have the labeled DNA. It's a ball of DNA. And in order to visualize the labels, we have to linearize. So this is done mechanically. This is actually done using electrophoresis as the DNA is forced into these nanochannel arrays. And these nanochannels are super small, so it kind of allows about 1 DNA molecule per channel. And as the DNA goes through, the optical part of the optical genome mapping, that's where it comes into picture. So there are these automated fluorescent microscopes or sensors that kind of take a snapshot of or image of these DNA molecules with the labels as they are passing through. So in this image, you can see that the labels are green, and you can notice the pattern of labeling in the backbone of DNA that is appearing as blue. So it not only captures these labels, but also preserves the information on how these labels are in relationship with each other, the distance between the labels. And that's what gives it the high accuracy for SV detection. I also want to mention that the optical genome mapping does it by imaging. There is also a technology called electronic genome mapping that uses a different technology to capture that information. Followed by this is the digital conversion of these images into digital molecules, assembly, and alignment. So first of all, before this happens, any of the only molecules which are more than 200 kilobase pairs, 200, 210, are selected. So anything less than 150 are filtered out. Any molecules with very few labels that are just going to be noise are going to be filtered out. So that's the quality check. And these molecules are then aligned to form consensus maps. And these consensus maps are further assembled to have a final assembly. And there are other techniques also that have emerged for the molecules to be clustered based on the SV to address the cancer SV detection. But I'm not going to go too much into detail. So this assembly, as you see here, you do not require a reference genome, right? These are generated based on the molecules itself. Now, after that, once the assembly is generated, you can compare this with the reference genome. So assembly is kind of de novo if you think about it. So once the final assembly, the sample of interest, is compared to the reference, the analysis is basically by comparing to how different it is compared to the reference. So there are different analysis pipeline. We used to have the de novo pipeline that was more appropriate for constitutional disorders because you could go down to 500 base pair level when it's present at sufficient almost heterozygous allele burden. But for cancers, the rare variant pipeline, where you can go down to 5,000 base pairs, but you can detect 5 to 10% allele burden. Now the guided assembly is emerging, which is mixing the advantages of both. Here is a point to kind of highlight the resolution of the OGM compared to the conventional carrier typing. It depends on the type of the aberration, of course, but you can see here for a typical unbalanced SV, like a deletion or an insertion, using the de novo assembly, it's actually 20,000 times higher resolution than the conventional carrier type. So again, compared to the other technology, the advantages and disadvantages of OGM. Now, OGM is a genome-wide technology, similar to conventional carrier typing, and opposed to FISH, which is more targeted. I'll let you guys go back and review the table here, but the most important thing is it does not require cultured cells, so it's particularly useful in cases where where you're not able to grow. And also, it can be, especially in cases of cancer, for example, lymphoid proliferation, this has particularly been useful, where it's revealed abnormalities that have changed therapy. And another point I wanted to make is, of course, unlike the chromosomal microarray, the OGM is able to detect balance changes, right? Balance translocations and inversions. So with that, let's move to how we evaluate the SVs detected by the OGM data. So here is, it's basically you're visualizing the data, generating the clinically meaningful report. The first picture or window to the data would be the circus plot. So as you see here, we have the ideogram. We have the ideogram as the outer rim. And the dots that you see here are the structure variants. We also have a copy number track. And depending on the application, we can insert the different bed files, depending on whatever application we want to use, to highlight those clinically pertinent abnormalities. So here are some of the structure variants that we can detect. So the first thing you see here would be the deletion. So first of all, the green lane is the reference, the green panel, and the blue DNA molecule is the sample of interest. And we can see these black lines. So these are the labels. And we can see the black lines from the reference connected to the sample of interest. Those are the match lines. Now, compared to the reference here, we can see that the initial labels match, but then after that, the labels kind of drop out. There is a few labels in red here that is highlighted in red are missing. And after that, the labels really go back to normal compared to the last label here. So you can infer that there is loss of some genetic material or a deletion. Using the same strategy, here we can see that there is repetition of a similar labeling pattern, the duplication. And then we have the insertion. We cannot really tell where it's coming from, but there is insertion. And then here, this is the classic hourglass match lines typical of inversion. And then, of course, here we see that the labeling pattern, to the left side match with one chromosome, and those to the right match with a different chromosome, so there is translocation. This is in cis, and this would be in trans. So these are the data that you can directly see. Then there's also information that you can infer based on the based on that data. So you can infer the presence of three-way translocation based on the breakpoints that are observed. Additionally, you can infer information on chromoanagenesis, chromothripsis, chromoplexy, chromoanasynthesis, all of that data routinely on any sample that we can see. Now, these are not the only kinds. The additional information that that OGM provides is also the copy number variation. Now, this is very similar to chromosomal microarray, but it's important that we kind of review this along with the other forms of data. So here is the copy number track that is visible at the molecule level data. as well as the whole genome plot, based on which we can kind of more accurately determine the whole arm changes or whole chromosome changes. The other key terminologies to use would be the coverage. That's, as you can imagine, the LOD is dependent on how many molecules you map. So on average, this can range between 200 for constitutional disorders to about 300 to 400 for cancers. The LOD for OGM is variable depending on the type of SVs, but on average is about 5 to 10%. Now, what are some of the challenges and limitations? Every technology has some challenges. There is no single technology that's perfect. So the technical limitations for the OGM is that we are not able to map the heterochromatin regions, only the euchromatin regions. The genomic regions near the telomeres and the centromeres cannot be determined. So this would affect the evaluation of Robertsonian translocations, for example. The P arms of the acrocentric chromosomes are generally not covered. And like with any bulk DNA technologies, the interpretation of high-level ploidy can be a challenge, such as tetraploidy or hyperdiploidy in BALL. So also keep in mind, beyond this, that when we are interpreting data from OGM, it's not just automated. It does require manual inspection. And based on our experience, there are certain difficult to map or polymorphic regions, especially clinical relevant regions that it's important to manually look for them, no matter what the color determines. This would be the BCL11B in ALLs, the IGH translocations because of the highly polymorphic regions, and CRLF2. And of course, collective efforts are needed to kind of meaningfully stratify these calls to those that are clinically significant, to those that are just variants of uncertain significance, what are germline, and especially with respect to the balance changes, more data are accumulating, and multiple center efforts would be particularly beneficial. So basically, the OGM is a very robust alternative to the standard of care technologies. And based on the advantages and the disadvantages, it can be very creatively used in conjunction with all other technologies for different applications. But it's also to remember when we kind of integrate different technologies together, it is important to remember the LOD of the OGM generally makes it a baseline evaluation technology, which is fine for constitutional. When it comes to hem-onc, it's not appropriate for minimal residual disease, although it's technically feasible if you massively increase the coverage. And of course, the throughput-wise, the constitutional, I said the coverage is of about 150 to 200x would be plenty sufficient, but for hem-onc samples, we need more than 300, almost 400x coverage. And again, we know it doesn't cover the single base pair level information, so especially for cancer cases, you need to supplement it with the NGS sequencing technologies. Also, it is important to keep in mind the cost as well as the accessibility of OGM. And when we are kind of integrating this in the context of other techniques, in addition to the clinical relevance, also to keep in mind the cost effectiveness that the OGM brings. Because it's a genome-wide technologies, panels and panels of FISH testing can be potentially replaced. So all of these come into picture. For example, in constitutional disorders, OGM would fit in very perfectly if the genome or the exome testing is negative. On the other hand, in heme, because it replaces the FISH that I'm going to highlight, it can serve very well as a first-year assay. So with that, we kind of, I wanted to highlight the potential clinical and research applications of OGM. There are several applications, and I'm only going to focus on 3 aspects because of the time constraints of the talk. So one thing to remember that the application in each field depends on the unmet needs of the field. and as well as the technical limitations of the standard of care assays available in the field. So for this talk, we can focus on some constitutional disorders for genetic diseases and quality control for the cells like the iPSCs, CAR cells, etc., and oncology applications. So the constitutional disorders, I think you must have, there are a variety of publications available in the literature as well as the sessions that highlight the value of OGM in identifying cases with undiagnosed genetic causes. It could be done as a reflex testing or as a complement to the standard whole genome or whole exome testing to evaluate the causes of infertility because of its potential to go down to a super high resolution. And many of the cases that are very challenging to map because the underlying genetic cause happens to be in the difficult to interrogate region, or any cases that might need further clarity or insights into the pathogenicity of SV, OGM can serve as a very nice supplement to the existing assays. And if there is a very strong suspicion of a genetic cause, but none of the other technologies have been able to figure out, OGM has shown that it has the ability to uncover the underlying genetic cause, and as is available in numerous publications. So in fact, this was a recent paper that showed beyond the exome, where the Greger Consortium proposes OGM as reflex testing for if the exome or the genome is negative. The second potential applications of OGM would be for the quality control. So for the iPSC, to assess the genome integrity in iPSCs or for CRISPR-Cas9 generation, or even for CAR-T. I'm just going to focus on CAR-T here because I have personally done these experiments, and I can highlight the value of OGM. So when we are generating CAR-T cells in the lab, invariably there is potential for introduction of off-target effects. So if it is because of the lentivirus, it can cause unnecessary genome modifications in sites other than intended. And the one way to potentially mitigate it would be to improve the T cell selection and to advance the vector design, et cetera. But a very simple way is to actually perform comprehensive genome testing before reintroduction of these CAR T cells back into the humans. So in addition to the sequencing, OGM, using a combination of both would provide a very feasible way to look at it before, to make sure that there is no damaging genetic changes that's already occurred. This is a proof of principle where we actually performed OGM on the peripheral blood cells as well as on the cells after the first introduction of lentivirus, and on the same cells after introduction of the second lentivirus with the CAR insertion. The first thing we found, of course, we were able to confirm the about 6.5 kilobase pair insertion of the CAR motifs into the cells, so that confirmed that it's actually indeed it worked. And we also noticed a couple of larger deletions that we do not expect. These would be the off-target modifications. And we're able to identify which area of the genome and the genes affected. We don't have much information on what these changes do, but as we do more and more testing, this is some valuable information that is super important for these patients so as to not reintroduce this. The CAR therapy is $1 million intervention, and this would be a very small cost for a quality check that could make a difference for the patient's life. And finally, I'm going to highlight the oncology application. So I'm a hematopathologist, so my presentation is kind of biased towards oncology, especially hemonc. Because of the easy accessibility of the sample the bone marrows and peripheral blood. We have plenty of it for cancer patients. And because the oncology has a super advanced approach, the precision medicine approach for which you need diagnostic biomarkers, prognostic biomarkers, as well as targeted therapy approaches, there has been a lot of advances of OGM in the field of oncology. It's by far the most applications, I would say. Now, just to highlight the need for OGM in this workspace, I'm going to present, based on the latest WHO and the ICC classifications, these are the categories for the acute myeloid leukemia with recurrent genetic abnormalities. As you can see, and don't forget the small table at the top too. So you can see, in order to accurately apply these classification criteria, how many genetic tests need to be performed on a single patient. Some of these are cryptic, right? KMT2A, MECOM, et cetera. So the number of FISH panels to implement these criteria can be massive. And this is not just acute myeloid leukemia. The same for myelodysplastic syndromes, where identification of any of the abnormalities to the right side could change the diagnosis. Let alone, I'm not even going to highlight the fact that the MECOM or the NUP98 rearrangement detection, which is majority of which are cryptic, could potentially open up the patient for enrollment into, say, menin inhibitor clinical trial. That's making waves right now. So, and again, this on top of the diagnostic criteria would be the prognostic stratification. For MDS, this is significantly based on the presence of mutation information, as well as the cytogenetics, the number of cytogenetic abnormalities, the type of structure, cytogenetic abnormalities, the presence of combinations of 2 or 3, et cetera. All these decide what kind of therapy the patient gets. The BALL, I don't even need to highlight here, just the fact that we can see here the pH-like BALL. We know that there are several fusions involved, and it is almost impossible using a FISH panel to identify these alterations. Now, each type of fusion responds better to different tyrosine kinase therapy, but we are at a phase where we are not even able to diagnose a pH-like due to the limitation in testing without a genome-wide approach. And of course, not to ignore the TALL, where there are several different rearrangements that are considered as different provisional entities for which different therapeutic strategies are used. So it doesn't take a lot to kind of know the value of a genome-wide approach for detection of all these abnormalities. And secondly, for consolidation of the lab workflow, right, to reduce the costs. I mean, performing a 20 probe FISH panel with 20 different probes, it's almost like we cannot even fathom the amount of resources that would be needed for that. And of course, these structure variants, in fact, data has shown that it impacts the prognostic risk stratification and also changes the management. So a lot of efforts right now, there is tons of data out there highlighting the value of OGM here, and the efforts are focused on how to identify how OGM fits in with the other existing standard of care workflows. So in fact, we've shown this with myelodysplastic syndromes. We did this on MDS just because it's a research interest to me. So this is a particularly difficult cancer to diagnose because it's a diagnosis of exclusion. You need at least a minimum of 40 different cytogenetic testing for cytogenetic alterations in order to apply the WHO criteria. For prognostication, we need complete karyotyping information, the number, the type, et cetera, in addition to the molecular data. And many of the MDS cases are now being treated using targeted therapy, like the TP53, et cetera. And we showed on about a series of 101 myelodysplastic syndrome patients that nearly 50% of the clinically important structure variants are not detected by the current standard of techniques. So this was distributed across a third of the patients, and not just because these were submicroscopic, but some of these are inherently cryptic, like NUP98. We found that MECOM was a particularly important alteration that was missed in about 80% of cases, and missed by both cytogenetics, and FISH was not triggered because there was no suspicion. And in our institution, MECOM rearrangement patients are enrolled into menin inhibitor trials, so it does have an immediate therapeutic impact for these patients. And overall, we showed about 97% concordance with the conventional karyotype. A couple of patients with non-informative karyotype had, with insufficient metaphases, we were able to demonstrate significant abnormalities. And overall, the most important thing we showed was the prognostic risk stratification changed using OGM data, and the data from OGM correlated better with outcomes. Now, this is just a single institution study. Of course, we need more validation from other centers, and that's already ongoing. And finally, I wanted to talk about the last point, the summary and future directions. So overall, as you can see, we can summarize that OGM is a very robust technology for detection of high-resolution genome-wide data on the structure variations. And we can go down, depending on the location, to either gene level or exon level. And these can uncover, in the hem-onc space, diagnostic biomarkers. It can consolidate the lab workflow and reduce costs, while at the same time provide high-yield clinically relevant information. And it's a powerful tool that is very feasible for routine clinical use, and it works with majority of sample material for the oncology space. And it can provide data within 5 to 7 days turnaround time, and that's very implementable for these patients. Overall, it enables application of the standard diagnostic and prognostic guidelines like ELN criteria for these patients, so we can practice at the highest level of care. And for the constitutional disorder, we've seen that the OGM is very aptly suited to generate a comprehensive and accurate cytogenomic data in conjunction with other existing techniques like microarray or sequencing, et cetera. So it has, as data has shown, it can uncover novel genetic abnormalities in constitutional disorders, thereby ending diagnostic odysseys for several patients. It can allow detection of repeat disorders like the FSHD, and detection of complex structural changes that are missed by even sequencing techniques. And it allows for detection of cryptic rearrangements as well. So, as I highlighted, the Gregor Consortium has highlighted OGM as a reflex test when the standard exome is negative. And finally, for research applications, as we've demonstrated, OGM can uncover, potentially discover novel therapeutic targets in the future. And it's useful for detection of it's a very simple, feasible, cost-effective technique to identify genome integrity for iPSCs, for CAR cells, et cetera. So that the impact of the OGM is actually demonstrated by the ISCN kind of quickly publishing the nomenclature. So there is standard interpretation and reporting across the world, so every patient gets the right amount of care. With that, I think I have left 5 more questions, but I wanted to kind of acknowledge a lot of people that are responsible for enabling my curiosity and experience with the OGM that started almost 5 to 6 years back. My clinical team in the Department of Leukemia, our Department of Hematopathology, led by Dr. Medeiros, a School of Health Professions, the master students from there who've done tons of work at the bench, the bio-nano team for guiding us throughout, listening to us and then improving the technology to make it clinically implementable, and the funding sources. And last but not the least, the International Consortium for OGM in Heme Malignancies. The founding members, we all started exploring the OGM technology together, and it was very nice to kind of share the pain and share the excitement when we were able to discover new things. So I'm grateful for that. With that, I'm open for questions.
Thank you, Rashmi, for this excellent journey through the field of OGM, its application, and it's wonderful that you have mentioned both the chances as well as the limitations. And I think there is a lot of things we have to learn, and you have called it pain as well as experience, and exchange is probably important. Therefore, we have some minutes now for discussions, so the audience is now.
Thank you for an excellent presentation. I'm Michael Gaddis, a clinician from Australia. Can I ask you to comment about normal variation you see in banding pattern in the population with this technology, and what's your policy on reporting variants of uncertain significance? Because I'm thinking there's going to be a lot of normal variation out there.
That's a very great question. Thank you for that. So the normal variation, I mean, for the copy number changes, I think there is enough data on that in the DGV and other databases. But like you mentioned, for the inversion, we've seen a lot of inversions, balance changes, that we are not really sure if it's a normal variation in the humans. At MD Anderson, we used the data from about 400 normal controls as reference. Of course, that's not enough, but as more and more data comes through, we will have more confidence level in calling. For reporting, though, I guess the VUS, it's different depending on the application. For the cancer space, we report everything we see, but we kind of keep it hidden as a tier 3, so we don't highlight it, and the oncologist can use the information as needed. But for the constitutional space, it might be very well relevant, right? So it's important to report the VUS if there's no data to dismiss that as a normal variation.
Next question, please.
Hello, my name is Valentina Ortiz from Hospital Niño Jesús. In Madrid, we use optical genome mapping for constitutional diseases in a clinical base. So can you comment about, on the deletions that are less from 100 kilobases? Because sometimes they offer only the boundaries of the deletion, but not the exact location of the little deletion. And it's very challenging because sometimes it's on a gene that you are interested about it, but when you do the MLPA, for example, it's not deleted that exon, so it's very challenging. Could you comment on that? Thank you.
Yes. So as I mentioned, like no single technology is perfect. In some areas, of course, the deletions, it might be challenging to interpret exactly like you mentioned. I guess the OGM kind of, at least it provides a clue to kind of verify that alteration using another technology. In the heme space, which is what we primarily use at MD Anderson, we have, for all the clinically relevant genes, OGM has been pretty accurate, both in our validation data and with our prospective experience so far. We have not had the need to doubt the call or at least for the validation, we verified all the calls, and since then, we've not had any discrepancy with other FISH or other techniques.
There's also a question online, of course. So could you maybe comment on the use of OGM in the detection of mosaicism? And I interpret this as how low can you go for mosaicism? And the second would be the use of OGM for marker chromosome detection.
Right. So in terms of how low we can go for mosaicism, in the cancer space, we have, so far at least, we've gone to the same resolution, as I mentioned, about 10%. So far, we haven't found any discrepancy. On a couple of cases, though, for the deletion 7q, for example, we have found OGM has missed it, but the cytogenetics has identified it in about 4 metaphases, and we interpret this as being due to proliferation bias, so it's been concordant. The other question was the marker chromosomes. Of course, OGM has a great potential to eliminate the marker, right? So long as it has the euchromatin. If it has only heterochromatin, of course, you won't be able to tell.
Thank you, Rashmi. Great talk again. Thank you.
Thank you.
We'll switch to the next talk.
All right. And it's a great privilege to also introduce the second speaker of this session. And before we start there is still some seats here in the front row. So please move in a little bit and all come to the front. There is really still some seats.